Welcome back to this second half of the afternoon. I just want to make one quick announcement before I hand over to Gabrielle. We have, as you know, a sociologist amongst us who's interested in how we collaborate, as John Tolan mentioned this morning. Now, I think it would be good for him if we were willing to share with him sort of our current status. So he's interested in looking at how collaborations initiate and how they evolve. And at the moment, he can't get the information without our permission to collect more information than just prospectively. In other words, he'd like to have a kind of retrospective view on on who we already know. Now, the director of the institute doesn't want to sort of put that upon us as organisers as a sort of burden to push out to all of you. But what I'd like to ask you, if you thought that this was a good idea, that you would share with him your current status of collaborations. I'm not exactly sure what his questions would be, but he would have a questionnaire. If you thought that that would be an idea you'd be willing to participate in, I'll ask you to raise your hand, but not just yet, because I want to say that, of course, nobody would be obliged to participate. So if you didn't want to participate, you simply don't fill in the questionnaire. Could you just now raise your hand if you thought that would be something you'd be prepared to engage with? Okay. Are there others here who feel that that wouldn't be a good thing for us to be doing? Is there anyone who thinks it's a bad idea? Nobody thinks it's a bad idea. Okay, so I'll speak to John Toland and say that this is what we think. And so you might get a questionnaire at some point. And as I say, feel free to fill it in or not, as the case may be. So thank you very much, and I'll hand you back to Gabrielle.
So welcome to this second part of the afternoon session. I'm Gabriel Valiente, another one more of the organizers. So you already met Water Geeks, and today you will meet the two more organizers of this program. First, Daniel Hudson from Tübingen, who will talk about pain of metagenomic analysis.
So this is joint work with Benjamin Bullfink, who's a PhD student in my group, and Jay Chow, who is a postdoc at NUS in Singapore. So here's an outline of the talk. I'll be talking about metagenomics, obviously, high throughput alignment, the DTRA challenge, and a new program called MALT. Okay, so metagenomics. This is when I first came in contact with metagenomics. This is a paper going back to 2006, where the main goal here was to see whether you can sequence mammoth DNA. And in any ancient DNA project, there's always contamination. So as a side project, we also looked at, what else is in the bone rather than just besides mammoth DNA. It was actually, I think, probably the first paper that used next-gen sequencing for metagenomics. And this was our result, a pie chart. Today, it'd be difficult just to publish a pie chart as a metagenomics result, but back in those days, it was a big deal. So in that context, I started working on a program called Megan, Metagenome Analyzer, that I've now been working on for, I guess, 8 years now. The code's becoming quite big and unmanageable, but still working on it. And then the only other metagenomics paper I ever was on was this one here, where we're looking at metatranscriptomics. And so even though I'm working on metagenomic methods for many years, I wasn't actually involved in any papers. And back in those days, like in 2008 and so on, I talked to quite a number of biologists in Tübingen, tried to get them interested in metagenomics, but there wasn't any interest really. And so things in Tübingen at least have changed, probably due to the Human Microbiome Project. So Human Microbiome Project looking at microbiomes in the context of human health. Now, just in the last couple of years, medical people in Germany got really excited about this. Now everybody wants to look at the microbiome in the context of some disease or something. And And so I'm involved in 5 different projects at the moment with different medical people. So I'm suddenly best friends with the Dean of Medicine in Tübingen, who I didn't know before, but so it's funny how that kind of thing happens. Yeah, so we're looking at a mouse model, a gnotobiotic mouse. So that's a mouse where you actually know exactly what bacteria are present, where you've got complete control over the gut microbiome and so on. We're looking at a mouse model for inflammatory bowel disease. We're looking at the result, what happens when when somebody who's obese goes through a diet or has some surgery to taking place at different treatments. We're looking at what happens when you take antibiotics. How do the resistance genes in your microbiome change? And different things. This is a typical also a typical project where you're looking at something, the interaction between Yersinia enterocolitis and the human gut cells. So people have looked at that as a two-way system where you've got the host and the pathogen, and now it's becoming a triangle where you're also trying to look at what are all the other microbes doing in this context. Yeah, so this is the kind of stuff that we're doing with the medical people. Things like you have a mouse that's originally not prone to getting inflammation, is prone to get inflammation, but if you change the microbiome in the gut using antibiotics and so on, you can actually get rid of the inflammation or vice versa. So by changing the microbiome in the gut, you can influence the disease state. You can influence the disease state. And so more recently now, interest has picked up in metagenomics as well. So I'm involved here in a number of different projects. Here I have a visiting professorship in Singapore where I work on wastewater treatment, sludge, and there's a number of other things. Ancient pathogens is quite an interesting topic. Ancient penguins is also of interest to some people, and so on. So, for example, in Singapore, we're trying to understand how wastewater treatment works. You think of the holding tank of the wastewater sludge as a huge organism consisting of many different microorganisms, and obviously you want to identify the species and you want to figure out what's going on in terms of metabolism and so on. And the ultimate goal is to improve water treatment performance. So Singapore gets a lot of its water from Malaysia, but would rather be self-sufficient if it could be. So there's a lot of money being put into this type of research. Okay, in that context, we've been looking at high throughput alignment. So there's a number of computational tasks that you have to undertake when you do metagenomics. We heard a lot about that from Volker this morning. There's binning and profiling. There's assembly, which seems not possible. And you want to compare different samples. And one of the things that we've been focusing on is just the old question of aligning reads against a database. And this is a computational bottleneck in many cases. For example, in the context of wastewater, we had a high-seq run of data that we wanted to compare against a RefSeq protein database. If you were to use BLASTX, we calculated that would take 29 years on a single multi-core server. So we didn't do that. We actually did this, ran RapSearch 2, not on a single server, but if we had done it on a single server, it would have taken us 7 months. And that's still not a very good turnaround time. If you've got a sequencer and you're sequencing every 2 weeks, you get a high-seq worth of data every 2 weeks, then you really want something that can run much faster. So we were looking at the question of how to speed this up. Okay, you're trying to do functional analysis. You've got a bunch of environmental sequences. So you don't always have a whole high-seq run, but a typical data set is going to be millions of reads. And the reference database depends what you use. If you use KEGG, that's quite small, but if you use the whole of NI, you're close to 40 million sequences that you're comparing against. And so first idea that we came up with was a method that we published at the beginning of the year called Powder. So you've got your protein reference sequence and you've got a read and you want to compare those two, and you know how that's done. You translate the read into protein sequence, and then you do an alignment. Interesting thing about protein sequences, protein alignment is that you're not looking for mismatch, but things that, like here, for example, in a T and a P, those are two different amino acids that quite often replace each other in an alignment, so you're allowed to match those. So the difference between DNA alignment and protein alignment is in the protein alignment you use a Blossom matrix or some scoring matrix to measure how similar, or to evaluate how similar two amino acids are. Okay, so powder, you have your protein reference sequence, you've got your read that you're comparing, and then it's just a very simple idea is you reduce the alphabet from 20 letters down to 4 letters. You can see I've already colored the amino acids there using 4 colors, and you get something that we call pseudo-DNA, and then you just take a DNA aligner. We looked at bow tie 2, do an alignment, and then you expand back to the original alphabet, score that, and so at least you get something, and that works really fast. Let's just talk about the reduction. So we looked at a paper here that was looking at many different ways to reduce proteins alphabets to different numbers of letters, and this is the one they suggested for 4 letters. And actually, if you take a bunch of BLAST X alignments, define a similarity score between amino acids, how often do they appear together in alignment, and then turn that distance matrix and run it through NeighborNet, then you get this picture here, and you can see quite clearly that these 4 groups that were described in a different context actually also pop out here quite naturally in a NeighborNet analysis. Okay, so that was our first idea, and then we did some benchmarking, and we saw that it runs roughly 10,000 times faster than BLAST-X. You don't see it here because here's only 7 minutes. 4 minutes of that was just to get the program started. Only 3 minutes of real computation. So for bigger data sets, it's about 10,000-fold speed up. But in this context, we're looking at comparing against the KEGG database, and you only assign 1/3 of the reads to a KEGG class that you compare to BLAST-X. So it's very fast, but it doesn't have the same sensitivity. Okay, so what you really want is something that's 10,000 times faster, but has the same sensitivity. Okay, we ran this on a whole high-seq run. Took a couple of days. Okay, but as I said, you only get 1/3 of the assignments. I don't know, Taxa or also KEGG. And if you look at how many alignments you get, you don't really get all the alignments. You get 2% of the alignments that BLAST-X would get you. So it's a nice first method, but it's not the answer to the problem of how to make that kind of analysis painless. And so I had a student, his project, Benjamin Buchfink, his diploma project was just to re-implement Powder in C++. We had done it in Java. We said, okay. So he went away, did that over Christmas, and then he wasn't really happy with Powder, so we just wrote his own aligner called SAS-X. SAS stands for sequence alignment using space seeds, and this thing's also 10,000 times faster than BLAST, but it's basically the same sensitivity. So we ran this on 29 billion reads, took 24 hours on a single server, and it was outputting alignments at a rate of 300,000 per second. That was how many it was outputting. So we ended up with 10 terabytes of output. So in a sense, we just moved the bottleneck along. So now you can quickly and easily compare reads against a protein reference database, but then the downstream analysis becomes so much more painful because then you've got all that data. Okay, so it's just a generic seed and extend aligner. Uses a hash table for the seeds. Methods like Bowtie use a fancy data structure, use a Wheeler transform to make the index small, but I mean a 500 gig of memory in a machine doesn't cost much these days, so it's not like you can't afford to buy the memory, so why save the space? Use a hash table, it's much faster. Space seeds are known to be superior over regular seeds and so on, in any case. So we've got a paper that's we're hoping to submit very, very soon. And so that was beginning of last year. And then I'd like to briefly talk about the DTRA challenge. So there was a challenge that started at the beginning of last year called Identify Organisms from a Stream of DNA Sequences. Here the challenge was to develop some software to identify organisms in a data set and also which genes. There was a 1 million prize for this challenge, and it started at the beginning of the year. We heard about it on the last day of May when the challenge was supposed to end. Nobody had actually been able to solve the problem until then, and so we took a look at this. Benjamin, J. Tao and I, we said it's a pity that we hadn't heard about this earlier. We could have actually taken part, but then we started working it nevertheless. And so here the goal is to identify pathogens in a mixture of clinical samples. So basically, in biothreat context, people start falling over and you don't know what's hit them, and so you want to do DNA sequencing then very quickly and very accurately determine what pathogens are being used. And so in this challenge there was data sets from 4 different sequencing technologies, and you were supposed to analyze them very accurately and also very fast. High accuracy, high speed and flexibility, being able to deal with different sequencing types of data. So this competition had 2 phases. In the first phase you could download 9 data sets, analyze them, upload your solution, then you'd get a score saying how well you're doing, and to enter the second phase of the competition you had to get 90% or better on each of the 9 data sets. And in the second phase of the competition you were asked to upload your solution, and then people looked at it and confirmed the performance and compared the speed to other good solutions. And so whenever you upload your data set, you got reports something like this. You've got an overall score which breaks down into 3 different sub-scores. Organisms, I guess that just reflects how many of the organisms did you correctly identify in the data set. Reads is how correct is your assignment of reads to the different organisms, and genes is obviously how did you predict the genes correctly. So here's just the 9 data sets that were in the competition. You can see, typical Illumina size, 6 million. There was some PacBio data sets with 2,000 base pairs roughly in read length, quite bad quality. So that was a bit challenging, how to deal with those. And so the approach that we used for this challenge was the usual approach. First of all, use a human-associated microbiome, so you compare against a human reference sequence to remove any reads that look like they're human contamination. And then we also looked at assembling the reads in between. Didn't seem to make any difference at the end of the day, so we skipped that. And then you have to assign reads to taxa, determine the genes. So there's nothing new here. So in this challenge, I guess it was really the details. How do you do this in detail? So the one key ingredient that you need for metagenomics, if you want to do it really fast and accurately, is have a fast aligner that's also very sensitive. And so, or can run in different levels of sensitivity. If you've got PacBio data, then you want to be very sensitive because of all the sequencing errors, but you don't have to be so fast because the data sets are not so big. And if you've got Illumina data, you can be less sensitive because there's less errors to deal with, but you have to be faster because there's more sequence. Okay, and so Benjamin took SAS-X, which he had written for a BLAST-X tool, and in a sense dumbed it down just to do DNA alignment, because aligning DNA is much easier than aligning DNA against protein. So this is a program that runs, this is the most comfortable configuration to run it on. 64 cores and 500 gig. That costs, a machine like that costs, a computer like that costs less than 10,000 euros, so it's not high end in any way. Yeah, you can take, so we took the whole of GenBank, the whole of microbial GenBank, and indexed that to produce a reference database, a DNA reference database, and run this program. This program is an all mapper, so it guarantees to find, well, within the range of the heuristics, it finds all high scoring matches. Not like Bowtie, which would find one or some of them high scoring matches, it finds all of them, which is important for metagenomics. Okay, so let's a bit hard. Pity I can't actually see the slides here. So we compare against the human genome, and the reads that don't match the human genome we compared against the GenBank. But one thing that we notice is that if you've got human-associated data, there's a problem if you're trying to call viruses, because there's a lot of virus and sequences in GenBank and synthetic sequences also that look like human data. So you're always going to get false positives there. So in addition to comparing your reads against the human genome, we saw that you actually have to also take your reference sequences and compare them against the human genome and then mask out stuff that looks like it's human. Otherwise, you're always going to have false positive virus matches. And then taxonomic analysis, we used the old classic LCA algorithm. You know, you have something that aligns to multiple reference sequences, and you place the read on the lowest common ancestor in the NCBI taxonomy of these sequences. This is the naive LCA. It's so a number of people in the audience have actually worked on improving the LCA algorithm. And the problem with the LCA algorithm, it considers each reading in isolation. It looks at read and looks at what it aligns to, and then it decides where to put that read. And so during this challenge, we looked at many different ways of improving this, and we came up with something that we called the weighted LCA. And so here's the basic idea. Here you've got your read, and so you've compared against a reference database. It hits multiple references. Now in the LCA algorithm, you would look at the species for each of these 4 references and go to the lowest common ancestor in the NCBI taxonomy. But in the weighted method, you take into account which other reads align to these different references, and then you place it above, say, 75% of the weight of these sequences. So here, this 75% or more of the weight of this is a much higher weighted sequence than the other one. The weight's basically how many reads match it. And so in this case, this read would be assigned specifically to the species of this reference rather than unspecifically to the lowest common ancestor. So that turned out to be a crucial feature of our solution to get the taxonomic classification correct. And then in gene calling in the challenge, that was quite a hard problem. So, you know, you're asked to provide a list of genes that you think are present in the sample, and a read will quite often hit multiple references, and the references will be annotated with different genes, and again you've got to make a choice. You can't report all the possible genes. You know, if a read hits 100 different reference sequences, you can't report 100 genes for that particular read. It doesn't make sense. And so here we use the weight of the references. So basically we ranked the genes by the weight of the references and took the top ranking ones, but we also took into account how much of a gene is actually covered, what percentage of a gene is covered by reads. So if you're the only read hitting a big gene, then it's most likely that gene's not the correct one. And so it's just a picture of this our solution for the DTRA challenge. It's called Metascope. I don't know why I didn't write it up here. And this will be available soon for academic use. Yeah, you compare against host genome, and then pass on the reads that don't match the host genome, pass on the count of how many things did match the host genome, compare against GenBank, and then do the analysis, the LCA analysis and gene calling analysis. And here we had 2 different reference databases, or 2 different versions of the GenBank. One just using SAS in its normal mode, and one using SAS in its so-called very sensitive mode, which we needed for PacBio and Ion Torrent data. Okay, so the challenge allowed you 1 hour for each data set, 3 for the Illumina data sets, and our solution actually does everything in about an hour. So that the problem, yeah, so that's all the dates. Yeah. So this was actually a cool day, the 11th of July, when our team was the first team to actually qualify for the second round of the challenge. So this was, yeah, and then into the last 4 hours of the challenge, we were the only team that had actually managed to qualify for the second round of the challenge. In the last 4 hours, 2 other teams just crept over the finish line. So then there was so we uploaded our solution to the challenge, and it took about 6 weeks until we got an email saying you have won the DTRA challenge, which was pretty cool. And it made big news in Singapore. I have no idea what this says. I hope it says positive stuff. And it was also, there was something in Science about this. Yeah. Okay. How long is my talk in any case? 2-4. Okay, 10 minutes. Okay. So I wanted to talk about something else. So briefly, this is a program that I've been working on just recently since the challenge. So as I mentioned earlier, Jay Tao and I came up with Powder as a way to do a fast protein alignment. Before we came up with Powder, we actually tried to write our own protein aligner in Java. It was supposed to be really fast and so on. It was ridiculously slow and absolutely no good. But after we went through the challenge, I actually saw that, yeah, you can do really fast alignment. And so I went back to my old code and resurrected it, and now I've been working on a protein aligner, or a tool that's going to be a companion to Megan. This is a tool that does, it's a fast and sensitive aligner. It does BLAST-X, BLAST-N, BLAST-P. Not only does it do that, you can have it output all the matches if you want, but it also does the Megan analysis on the fly. So if you don't want, you know, huge files with alignments in it, you can just have the final analysis. Okay, so you get a file that's small, but it tells you exactly these are the taxa I found, these are the genes that I found, and so on. So yeah, I don't know. I don't have to say much about that, I guess. So it's similar ideas to SAS, two programs, one that builds an index and then one that runs the actual program. And it produces alignments if you want them in SAM format, but it also produces a Megan file that you can load directly into Megan. So for people who, we heard this morning about MG-RAS, and a lot of people use that resource, but for some people are protective of their data. They want to keep it in-house until they've done the final analysis. So for people who are interested to do that, they could just use Molt and Megan together, skip the part where you output huge files containing alignments and go straight to the part where you've got something that you can look at in Megan. And there's all kinds of things that you wonder why they're not in every program, like the reference sequences can be in multiple files. The files can be zipped. The input files can be multiple files. They can be zipped. You can have everything zipped coming out and so on. So just a bunch of useful, scientifically not interesting, but from a practical point of view, very useful features implemented there. So this is our workhorse at the moment in my group. We do all our projects with this. So we use it to compare against DNA against GenBank, proteins against NR and RefSeq, and we also use it just for doing 16S analysis. Take your whole shotgun DNA sequence and just align it against silver. Then all the 16S sequences will pop out, and you have a 16S analysis. If you've got millions of reads, 1 in 2,000 reads is going to be 16S. So that gives you quite a lot of 16S sequences that you can use. So I've just recently been studying the properties of this program using some data sets from the Human Microbiome Project, some mock communities where they produce some data sets by taking different, sorting cells together and then sequencing them. So this is, as Volker pointed out, this has got nothing to do with real metagenome data sets, because they've got 25 species and they're clonal, and no real world data set looks like that. But on the other hand, you could say if you can't even analyze these data sets correctly, then you've got no hope of analyzing more complicated data sets. In any case, here's just some benchmarks. So for 1 million reads, this is not as fast as SAS, but it takes 30 minutes to compare 1 million reads against the whole of NR, 10 minutes to compare against the microbial GenBank, and 2 minutes to compare against silver. And I've been looking at plots of this nature where the black line tells you what's actually in the sample, in this mock community, this cell sorted community, and then this is what Megan actually assigns. So here you can see there's quite a big discrepancy between the number of organisms in the community and how many reads get assigned to each of the different taxonomic nodes. And the horrible thing is that you always get this long, long tail of stuff that's not there. You know, people get intrigued about how diverse the environment is and so on, but even, you know, a lot of that might just be artifact of methods. It gets, the worst kind of problem is when you're using NR to analyze a data set. If you're doing a protein alignment, okay, so here again, this line here tells you how many reads, how many organisms are in the data set. You know, mostly 100,000, and you can see this is how many reads get assigned to those different organisms. But then you see this tail. There's 23 genera in this data set. We know that, but the analysis gives you 700. So that's a huge discrepancy. I mean, we saw numbers like that in Focus talk this morning. That there's orders of magnitude of difference between, in this case, between what's actually in the data set and what different analyses are finding. So at the moment, this is what I'm working on at the moment, trying to get away from 700, cut the number down to 23. I mean, some people in the audience have published papers in the past showing that, you know, Megan has problems, and this is one of the problems that people mention, and now I believe those guys. It is quite scary. Okay, just some conclusions. Basically, in any case, this program will be available very shortly. The paper is being written, and this is also, I mean, if you're interested in this, people who are interested in trying out mold, I'm happy to share that immediately with people. This will take a bit longer. Okay, and thank you very much for your attention.
So, isn't assembly the answer, or at least partly the answer, to both the issues of speed of taxonomic functional assignment and specificity that you mentioned at the end?
Speed. So for the DTR8 challenge, we had two solutions. One that we use assembly, and one that we didn't. We actually took the assembly, and for the purpose of the challenge, at least, there was no difference. We could do equally well, and we took the assembly out just because that's for assembly, every now and then the assembler just took way longer than you'd expect it to take for unknown reasons. So it's an unknown in the equation. So we took that out. And I mean, Focus talk this morning showed us that assembly is not the answer, because you get those. Yeah, he just showed us a profile. This is what the reads. Maybe this is showing that. Yeah. Any case, Focus showed us there's lots and lots of different taxa. If you trust the reads on their own, there's only very few taxa you do assembly. Maybe it does go in that direction. Maybe you're right.
That's my experience anyway.
Okay.
I mean, I've used Megan a lot and like it because it's one of the only things that can really let you explore the data rapidly. But what I would much prefer to do is assemble my data set and then put it into Megan and then explore it that way. But at the moment, that's a bit difficult to do.
Okay.
On you're looking at your mock community there, how much of the problem is the alignment rather than the classifier? Do you know if you ran that's with your aligner? If you ran that with BLAST X, would it look better?
I didn't try, but I would. It's 99. I'm pretty sure it's not the aligner. It's not. This is not an artifact of the aligner. You get exactly the same if you did BLAST X or anything else. So the aligner doesn't. It's not generating artifacts. It generates. I mean, I've debugged the aligner. It's not generating garbage alignments, and the alignments have BLAST. They have E values and so on. I'm only taking the high ones. And
when you're comparing your benchmarking to, you say the sensitivity was the same as BLAST or similar to BLAST. What kind of comparison? That's a mock community to calculate that?
No, that's where you just run the program, the two different programs on the same data set, and then you match up the alignments. Just count. So that wasn't a systematic study that we took some. Yeah. So for that we just took a data set that we've been using a lot, permafrost data from one of Janet Jansen's publications, just because that's what we had BLAST matches for. We just compare them and we list the alignments and match them up.
A deeper level, the solution to that problem is some kind of mixture modelling, where you say, if we have 700 species, this is what the data would look like, and I bet it doesn't look like that.
Yeah. Okay. Yeah. I mean, when that sounds like the kind of stuff that we should be discussing in this program here, huh? Like more mathematical, like model-based analysis, not just blast and.
Yeah. You can do all the assignment of read by blast, but then you say, when you fit the jigsaw puzzle together, does it actually look like?
Does it look like something that would come from 7 other species? Yeah. I think Fock has been.
Yeah, a couple of comments. I like the fact that you mentioned that your data exploded after analysis, because this is typical for metagenomic analysis. You mentioned that you went from gigabytes to terabytes in terms of file size.
10 terabytes. We couldn't even. The way that we actually did is, we did some of the data, then we got the stuff and we deleted it. We didn't have 10 terabytes.
So we see this a lot. So typically we see a 10x explosion on disk, and it's getting worse. This makes metagenomic analysis a unique sort of pipeline, because most of the other pipelines go the other way, right? You compare to the human reference, and suddenly your gigabytes are, a few megabytes. So that's number one. The other thing is, you mentioned running analysis local, and I think that there's a lot to be said for that. I just want to point out that increasingly reviewers will ask, how does your data set compare to X? So you're entering a little rat race there, right? When you just finish analyzing against the first round of reviewer comments, which usually takes a while, there's new metagenomes out there, and this will go on and on and on. I think in the end, we need a community solution for that. Either tell reviewers, don't ask me that, or we need pre-analyzed data sets that you can search against, because otherwise what will happen, and it's even worse than that. You mentioned that you have multiple different databases. So I've done my analysis against, I don't know, Seed, Keck, what have you, and the reviewer really is in love with one specific database. The next thing you will be asked, so how does it work for, I don't know, Go or Knox or whatever. So there's a huge problem out there. And again, that we need to have a community level solution for that, or else you're screwed, because it's an
But that problem's just like in phylogeny, isn't it? Whatever method you use, the referee's going to say, why didn't you use Bayesian? Why didn't you use this and that?
I agree. I mean, you can spend so much time on comparing against other tools, and there's always the next tool coming out, so there should be some convention to compare against the two best tools at the moment, which have some different aspects, and that's it. I think one should also agree when looking at papers to not ask people to look at 10 tools in parallel, because you'll never get anything published that way.
So we're hoping that when we publish the Metascope paper that we say, okay, this won the challenge, so that's got to be proof enough that it's a good tool. We'll see whether the reviewers go along with that, or they say you still got to compare against X, Y, and Z.
Provide some data that supports that statement. I mean, are the results public? Or?
So I think the plan is to have 2 papers back to back. The DTRA people are going to publish their pipeline and their evaluation scheme and so on, so that would be of interest to you, because you're thinking along similar lines, aren't you, to do some kind of community benchmark thing.
Right.
Yeah. And so hopefully there'll be their paper saying, okay, this is how we scored things, and there'll be the paper describing the method that won the challenge.
Yeah, one more add-on. We find a lot of people are using Amplicon in addition to Shotgun.
Yeah.
The prices have gone down so dramatically that for $15 or $20 you can do the Amplicon analysis. So a lot of people sort of back up there, whether it's assembly or clustering based stuff like the 16S Amplicon. So I would personally view that as something that would be just a kind of requirement for publication in the future. If you don't have the $15, it can't be something you're really interested in.
More questions? So let's thank Daniel again.